All right, good morning. I hope you have drawn your picture. This is the picture that I drew from the book, but it might be a good idea for you to grab your textbook and have your textbook sitting next to you. We are in chapter 15 now, and the picture that I had you draw came from a combination of things on page 304 and 305. So if you'll grab that real quick and we can get started. You also want to have your own personal picture out, but you want to have some notes out so that perhaps if you need to write some extra things and you don't want to mess up your paper, then we can work on that. All right, before we get started, I do want to say that this textbook or workbook is probably a good idea for you because this will help you review for all your stuff, and you can have it handy in front of you for the day of the exam. It has all the different topics. Obviously there's some things in here that we won't need to know, but it does go back through all the labs that we've done. So here's something about water potential. We've got some photosynthesis. We're going to do that next week. Mitosis, that wonderful mitosis lab that we did. It has some DNA fingerprinting. It has our animals, their choice chamber. It has enzymes bubbling. So these are really good for you to have because your first question on your exam is about one of the labs or an experimental design. We'll be reviewing about how to graph things and make error bars and whatnot. You won't have to graph, but you'll have to understand them. And so if you don't even know how to graph them, it doesn't even matter. You won't be able to interpret what's going on. So we will be working on that as we get closer to the exam. But if you could order one online used, and you might as well go ahead and do that quickly because a lot of those things take a week or so to come in. This is going to be something that you'll have really handy right in front of you that has all the main information. Okay, so first thing we're going to talk about is turning things on and turning things off. The name of this chapter is Regulation of Gene Expression, and what it stands for is we don't want genes on when they're not supposed to be on, and we don't want genes off when we need them to be on. And if you remember, I'll write this down so you can see it. Everything we've been talking about is genes and proteins and transcription and translation and all kind of vocabulary words. So in your DNA, okay, you have segments of your DNA called genes. And genes code for a couple of different proteins depending on the way it's edited. And then those proteins are what make you. So those can be enzymes, muscles, hair, skin. It can be parts of the ribosome, even though ribosomes are doing all these processes, okay? So the whole deal is your DNA's in your cell. It has genes to make proteins so that you can live. If something in any of these steps is messed up, then these things are going to be mutated. And so we want all of these processes to happen like they should. But another thing is every single cell, every cell in your body except for the egg and sperm has all 46 chromosomes in it. And so in your heart versus your kidney cells, so heart cells, kidney cells, and we'll go with, I don't know, pancreas. They all do different things, okay? But they all are made of cells from you that have all the exact same directions. So they have to figure out which genes do we turn on and which genes do we turn off, okay? Kidney on and off, and pancreas on and off. Okay, and if they do it wrong, then you have issues. But 99.9% of the time, all these cells are doing the right thing, and they have the right ones on and the right ones off. Okay, the way they turn genes off is they methylate them. So they attach clusters of methyl groups to the actual chromosome and essentially glue the pages shut so that you cannot turn the genes on in the heart that the heart does not need. Okay? But the kidney cell might need the genes that the heart cell methylates. So the kidney cell will leave those unmethylated and they will methylate different genes depending on what's necessary for them. Same thing in your pancreas, same thing in your skin, same thing in your brain, same thing in your eyes, okay? All of those cells have the exact same chromosomes, but they turn different ones and different ones on and off depending on what they need. And that is regulation of gene expression. Gene expression is the fancy way of saying turning genes on, turning genes off. Regulation means to control it. All right, so we have two types of cells in the world, essentially. We've got prokaryotic and we have eukaryotic. Prokaryotic cells are very simple. They have no nucleus. Okay? Eukaryotic cells are complex. They have a double membrane-bound nucleus. Okay, so they have all their DNA very protected inside that double membrane. Prokaryotic cells, they do have DNA, but it's just, if you look inside the cell, sometimes it looks like a little squiggly. I think we call it like a little spaghetti. And then sometimes we have these round little pieces, and we learned about them a little while back. And these little round pieces are called plasmids. Okay, still made of DNA, and they're just in a circle. So it's hard to figure out where to start and where to end because a circle has no beginning and no ending. Eukaryotic DNA is in the form of long strands called chromatids. And then it's made of chromatin, and then usually you have two of them bound together whenever the cell is ready to divide. So it looks like an X. So we got noodles and circles and X's, right? Prokaryotes have to make proteins, so they have ribosomes. It's have to make proteins, so we also have ribosomes. All right, and the process that is made in the ribosomes is called translation. Translation in both places. All right, so prokaryotic cells take the DNA, turn it into messenger RNA, and that's called transcription. Transcription we've talked on and on and on about. So the piece of DNA turns into messenger RNA, and the messenger RNA goes to the ribosomes, and the ribosomes does translation, and it makes a protein. Same thing here in eukaryotic cells. The DNA goes to pre-messenger RNA, makes a piece of messenger RNA, we edit it, we take out the introns, leave the exons, we add a cap and a tail and all kind of stuff. We're super specific and very careful in eukaryotic cells. And then we get our messenger RNA, and then it goes to the ribosome and it does translation, and we get our protein. Okay? And this is going to happen every time the cell is signaled to make a certain protein. All right, but the human body, the plant body, the bacterial cell, all organisms are super efficient, and they are not going to waste any energy keeping things on or off if they don't have to. All right, so the same thing happens at your house. And in your house, we're going to have, oh, it's a beautiful light bulb. Okay, so you get the light bulb. All right. And you walk in the room, you turn it on so that you can see. And if you're lazy, you walk out of the room and it stays on, and Dad is not happy, right? So mom's not happy, dad's not happy, I'm not happy because it's cost a lot of money. All right, so in the cell, when things are made or used or processed that aren't necessary, they don't spend money, but they do spend something that's called ATP, and that's their energy. And if they're spending energy when they don't need to, once again, frowny face. It's stupid, it's inefficient, and it's a poorly made organism. So the most complex organisms, they turn genes on and off only when they need them. And even the simplest bacteria cells turn genes on and off only when they need them. Okay? And we're going to talk about how we can turn things on and off. All right, so the first one we're going to start with is prokaryotic cells. Okay? So I'll read this to you. You can take notes and write this down and put the answers in as we go. But it says when anything binds to a protein, and this is like super important. When anything binds to a protein, it changes the protein. Okay, so when anything binds to a protein, it changes the shape of the protein, which changes its function. Now when I say it, I'm going to write changes the protein's function. Okay, and so right here, structure equals function. Okay, this I can't overemphasize how important it is that you remember that the way something is shaped or looks is completely related to its function and what it's supposed to do. All right, so the next sentence says, houses usually have a light source in every room, but it would be a waste of energy to leave every light on all the time. So there are switches to turn the off the lights when they are not in use. Okay, and that makes sense, and everybody knows that, and you've probably been yelled at for it. All right, sometimes one switch controls several lights in the same room. All right, and that's kind of cool. So not all houses are wired that way because it's kind of, I mean, sometimes you don't want every single light on, but in my kitchen, I have a switch and I have lights in the ceiling, and I can turn on, it turns on all the, like 3 or 4 lights at one time. But then in other rooms, you just turn on a switch and it turns on one thing. So depending on what you want and the purpose, it's easier in my kitchen to turn on just all 4 at once than it is to go around and have like a separate switch for each light. But in my living room, I don't want all the lights to come on at one time. So this is the point I'm trying to make is prokaryotic cells can turn on genes, can turn genes on and off based on environmental factors, okay? So environmental factors, it does not have to be rain and temperature and weather. Environmental factors can be that something is there or something is missing. It can be temperature, and it can be precipitation. It can be acids, in which would make them the pH change. But any type of environmental factor would make the genes turn on and off. And sometimes related genes are grouped together with one switch. So that's like what I was talking about in my kitchen. Like all of the lights, recessed lights in the kitchen, I can just turn them all on with one switch. This group of genes, along with the section of DNA that regulates them, so there's a section that can regulate if the genes get turned on and off, it's called an operon. Okay, and I need you to overemphasize this is prokaryotes only. Eukaryotic cells only do one gene one at a time. They do not turn genes on in clusters. All right, so there are lots and lots and lots and lots and lots of operons in prokaryotic cells, but we're going to do 2 main examples, and they will be your 2 go-to examples for the 2 categories, okay? So we have inducible and we have repressible, okay? So inducible operons are ones that are off, but we can induce them to turn on, and then repressible is that they're always on, but we can actually turn them off. Okay, so first one we're going to talk about is the Trp Operon, okay? It is always on. and it's always on because it makes something for the prokaryotic cell called tryptophan. And tryptophan is extremely necessary. And so if it's not on, then it doesn't make tryptophan. If it doesn't make tryptophan, the prokaryotic cell will die. All right, so the only way that this thing will turn off is if there's so much tryptophan that it doesn't need to keep going on. Okay, so the Trp Operon is always on, but it can be turned off. Okay, so things that are always on but can be turned off are called repressible, because we can actually repress them, but that means that you got to turn them off. They're going to stay on all the time. And in your house, that would be your refrigerator, that would be your heat and air, that would be your hot water heater. You want them to always be on and only turn them off when necessary, okay? So I'll put fridge. The time you would turn the refrigerator off is if you have to clean it out and remove it. Otherwise, you're gonna leave it on all the time, okay? Over here, our second type of operon, or category of operon, let me change my pen, is, we're gonna call it the lac operon. And lac stands for lactose, okay? Lactose is a sugar, and it has some cousins that have similar shape and function, so allolactose, galactose, some different things. So if a bacteria cell comes in contact with lactose, then all of a sudden it's like, oh, I need to break this down, I can use it for energy. So usually it's always off, okay? Because there's no point in making lactase, okay? So that's what your lac operon does, is it makes lactase. So there's no point making lactase if there's no lactose, so it just chills, and when it gets exposed, environment influence, so it gets some sugar in its environment in the form of lactose, it needs to make lactase, so it's going to take this operon that's always off and turn it on. So this is going to be called inducible. All right, so we can induce it just like you induce someone to go into labor, and I think that was one of the questions I had on your assignment yesterday. So when you induce someone to go into labor, they're not in labor, so you're giving them something to make them go into labor. If you induce a scenario, you make it happen. If you repress something, you keep it from happening. So these are your two main operons, and operons look like this. And this is your picture, okay? So they're gonna have a section up here that is a regulatory gene at the beginning of, so this is just like a section of the DNA. So this is the 3' end, and over here you're gonna have the 5' end. And so this is just a segment of DNA, because genes are sections of DNA. But the operon has a gene here, and then 1, 2, 3, 4, 5 genes here for the Trp Operon. All right, so it literally can turn on 5 genes at one time with this one promoter switch. So here's our light switch that's going to turn things on and off. Now right now, that is your light switch. And right now, this egg-shaped bubble is our RNA polymerase. And RNA polymerase's job is to do, okay, so that RNA polymerase is going to move all along this piece of DNA and make a copy of messenger RNA. And that copy of messenger RNA, the process is called transcription. And it doesn't just transcribe one gene, it transcribes gene E, and it makes this piece of messenger RNA. So it has a start codon, and then it has a stop. But then immediately after the stop is another start, and it makes the piece of messenger RNA for gene D, and then C, and then B, and then A. So rather than doing each one separately and wasting a lot of time and energy, it makes all 5 pieces of messenger RNA on one strand. And then that piece of messenger RNA will go to the ribosome. And the ribosome will start and stop and make polypeptide E. And then it reads and it starts and stops and makes polypeptide D. And then it makes C, and then B, and then A. Why they're backwards, I don't know. I don't know why they're named that way, but whatever, okay? So these are your products. So these are your proteins that we're talking about being made. And the reason they're being made is because they have to be used. They're actually enzymes that join together at certain points, and they turn this precursor molecule, pre means before, into, after a series of reactions, tryptophan. And tryptophan, I told you before, is extremely important to the prokaryotic cell, so it's a necessary component. So the 2 proteins made from gene E and D actually join together, and they make this enzyme. And the enzyme, thankfully, is named enzyme 1. And enzyme 1 catalyzes the first reaction. So it takes this precursor molecule that's floating around in the cell and catalyzes it into this shape. So it changes the shape. Then the protein that's made from gene C turns into mRNA-C, and then it makes this polypeptide protein, is called enzyme number 2. And enzyme number 2 catalyzes the second step. So 1 and 2 join together, E and D join together to make enzyme 1, and then they get this. And then this enzyme catalyzes the next reaction. And now we have gene B and A, which make polypeptide B and A join together, and they make enzyme number 3. And enzyme number 3 catalyzes the third and final reaction that actually ends up making tryptophan. So if anything is messed up along this route, this step may not happen, this step may not happen, and this step may not happen. All steps are necessary to make tryptophan. So this whole gene series, so 1 gene, 2 gene, 3 gene, 4 gene, 5 gene, 6 gene, all is necessary to get all the way to the final step, which your ultimate goal is here, tryptophan, okay? And tryptophan is one of your essential amino acids that we need. All right, so here, this is where kids get a little bit confused. This gene is called a regulatory gene, and regulatory genes regulate. So what you see here is the regulatory gene also makes a piece of messenger RNA, also does transcription, and then it makes this product, okay? It's super important because this is your repressor So a repressor protein is a protein that can stop the whole process from happening. And he can actually come up here and bind to this promoter region. And see how the little puzzle piece has these little pieces. But right now, because he's bent in a funny shape, he doesn't fit in there. And so the fact that he doesn't fit in there means that this RNA polymerase can just run straight down this strand and do the job, okay? So when nothing is there, this operon is on, okay? And that's the way it likes to be. It likes to stay on. But sometimes things happen, and we can actually turn this operon off. And the way we turn this operon off is this guy right here. And this is tryptophan. Now tryptophan is what was being made from this whole operon. But this is supply and demand. And the supply being made is tryptophan. But if the prokaryotic cell decides it doesn't need it that much, then the supply increases so much that there's an excess. And if there's an excess and it's not being used, then it will actually bind to the repressor protein. And when it binds, anything binding to a protein changes its shape. And when it binds, well, we'll also say it changes its shape. And now this can actually move up here and it fits. And because it fits, it blocks the RNA polymerase. Okay? So when the RNA polymerase is trying to move down this strand and do the transcription, it's stuck. It can't get there. The only way this can turn back on is once the tryptophan levels in the cell get too low, steals the tryptophan repressor protein, this changes protein back to its becomes unbound the operon is back on. So right now, because this tryptophan has bound to the repressor now can fit, and this whole thing is off. And the only reason it's off is because the tryptophan levels got so high that the cell doesn't need to make any more for a little while. But once the levels get low, it'll start breaking this down and using it, and then that thing won't fit anymore, and then you're gonna get this, and it's gonna be back on again. So it'll make tryptophan and make tryptophan and make tryptophan. But then, too much tryptophan, it can turn itself off by binding. So this is this actually, it regulates itself. So when there's too much of it, it can turn itself off. And when there's not enough of it, it can turn itself on. All right, so to summarize, Trp Operon. Trp Operon makes what? Makes reactions. Okay, so we're gonna we can be a little bit more specific and say it makes enzymes, which catalyze 3 reactions that turn a precursor molecule into what? The whole purpose of the Trp Operon is to turn it into tryptophan. Okay, and tryptophan is necessary, so you can think of it as the gold. All right, so a Trp Operon is always on, and it's because the bacteria always needs tryptophan. Excess of tryptophan, the excess tryptophan can actually turn the operon off so as not to waste what? We're going to say energy. No point, all right? This makes tryptophan a blank because it must bind to the what? Okay, so here is the cool thing. Tryptophan binds to the repressor protein, so it helps it out in its job. And so we call this a co-repressor. Okay, so tryptophan is not the repressor, but it's a co-repressor because it makes the repressor able to work. All right, so this makes tryptophan a co-repressor because it must bind to the repressor shape of the repressor so that the repressor can now fit into the, there's your yellow site, so your operator. RNA polymerase from moving down the DNA? What's the answer there? Block. Good. Your operator's here, your repressor protein's the wrong shape, it can't fit. So when it's not blocked, it means that the Trp Operon is on. When you have excess tryptophan, supply and demand, we don't need to make any more. So that excess can now fit into the repressor protein, which changes its shape. So now instead of being curved and disfigured, it's nice and flat. It slides right in there and it blocks it and nothing happens. So when this is blocked, it means the operon is off. Okay, and because tryptophan has to bind to make the repressor work, tryptophan is called a co-repressor. All right, so I hope that makes sense. Go back through. You can pause the video if you need to, but that was your little introduction to Trp Operon, and tomorrow we'll do lac operon, and then we'll talk about how eukaryotic cells do the same job. So have a fabulous day, and just remember, they're on when you need them, and they're off when you don't need them.